animal interface and also way forward. So influenza is highly contagious acute respiratory disease caused by influenza virus. Virion or virus is irregularly shaped spherical enveloped particle. It belongs to family Orthomyxoviridae. I'll just put a and size of the virus is 80 to 100 nanometer in diameter. Genome is single stranded negative sense RNA in 8 segments. Why I have made this red is because this is the reason why there are reassortments, as Dr. Nivedita madam just explained. So genome is divided into 8 segments, negative sense and RNA. So it is there are possibilities of reassortments between human influenza viruses, animal influenza viruses, avian influenza viruses, and it has been shown in the past that this is how pandemic strains were generated. Surface proteins are important. HA is hemagglutinin, which is used by the virus to attach to the host cells, and antibodies to HA protein are also protective. Neuraminidase is another protein which helps virus to release from the host cells after the last stage of infections, and majority of antiviral drugs are being targeted against neuraminidase protein. Currently available oseltamivir phosphate is also antiviral drug which is targeted against neuraminidase protein. So as far as natural host of influenza viruses are concerned, avian, horses, that is equine, then pigs and humans are reservoirs. I mean, they are the host, natural host of influenza viruses. There are 18 subtypes of HA and 11 subtypes of neuraminidase surface proteins. So 18 into 11, 198 possible subtypes are possible. And if you see here, all the HA subtypes, 16 HA subtypes are present in birds, 9 are present in birds. So this influenza is basically avian virus, and during the course of evaluation, these viruses have adapted into human beings. So AI disease in birds is classified into 2 forms. One is highly pathogenic avian influenza virus, which causes sudden onset and mortality can reach in birds up to 100%. Another form is live low pathogenic avian influenza virus, which causes mild form of disease in which there will be ruffled feathers, reduced egg production, mild effects on respiratory system. As far as reservoirs of avian influenza viruses are concerned, birds from Anseriformis, in which ducks, geese, swans are there, and then birds from Caridiformis family, in which gulls, terns, and waders are there. These are natural reservoirs of influenza viruses. So talking about the outbreaks, the first HPI H5N1 outbreak was reported in Maharashtra in the year 2006, and there are reports that genetic or molecular analysis showed that these viruses were similar to viruses from Italy and Iran. Then there were samples collected from human beings who were exposed or who were involved in culling, and samples from those exposed humans were negative for H5N1 virus, indicating there were no human infections. So initially, all the outbreaks, majority of the outbreaks were focused in the northeastern region, and then currently globally more than 70 countries have reported outbreaks of HPI avian influenza. And as far as India is concerned, 24 out of 28 states as well as 7 union territories have reported outbreaks of avian influenza H5N1. And these are the different clades of H5N1 viruses which have been reported from India. So this is one experience which I would like to share, that earlier outbreaks were focused in poultry, but in 2011 there was a widespread mortality in crows in Jamshedpur in the year 2011. Approximately 4,000 crows died, and initially it was thought that it probably could be West Nile virus, which is another virus which can cause mortality in birds or crows. And this virus, this actually deaths were due to HPI H5N1 virus. As far as human-animal interface studies or screening of humans are concerned, all the humans who were exposed were screened and they were negative for HPI H5N1. So till this point, till 2016, all the outbreaks were due to H5N1 virus. But in 2016, there was unusual mortality reported in migratory and semi-feral birds in National Zoological Park in New Delhi. So this was the first report or occasion that another HPI virus, which is H5N8 virus, caused mortality in bird, and this virus was found to be similar to virus A duck, Eastern China. and it had a avian receptor specificity, and there were no human infections here. So later on in the year 2021, there were outbreaks caused by both H5N1 and H5N8 simultaneously. So in Kerala, virus was H5N8, and there was mortality in ducks. In Gujarat, it was H5N1, and in Maharashtra, it was H5N8. So again here, screening of humans showed samples were negative for avian influenza, as this was during the COVID-19 period. Some of the samples were positive for SARS-CoV-2. So then we asked a question, because there was no data on seasonality of outbreaks in our country. So we perform spatio-temporal distribution and seasonality of HPI, H5N1 and H9N8 outbreaks in our country from 2006 to 2021. And what we found that maximum outbreaks, I mean, since the first outbreak in 2006, all the years outbreaks were reported. Maximum outbreaks were reported in the year 2021. And currently, as Dr. Nivedita Madam explained, currently outbreaks are ongoing in the February in the state of Andhra Pradesh. Then year-wise analysis showed that maximum outbreaks were in the winter season in January, and this actually coincides with the migratory bird season. So this data is important as far as seasonality of avian influenza outbreaks are concerned. So we performed studies on evolution, because there are not many studies on phylogeography and gene pool analysis of HPI, H5N viruses from India. So our findings show that there are 8 reassortant genotypes which were circulating, and there is inter-subtype and interclade reassortments. That means there is exchange of genes between different subtypes, and these viruses are were found to be circulating during these outbreaks. As far as phylogeography is concerned, there are transmission links between East Asia, Southeast Asia, India, East and Middle East Asia. So this shows transmission links between of H5N1 in this region. As far as low pathogenic viruses are concerned, there are reports of H9N2, which is a low path virus, H4N1, H11N1, and a few more viruses from our country. So here, this is the scenario in which shows H9N2 prevalent in poultry as well as in wild birds. So we also studied evolution of these LPI viruses in our country, and our findings show that the virus H9N2 in our country belongs to classical G1 lineage, sublineage Middle East B, and there are 5 genotypes which were circulating, and there is again inter and intra subtype reassortment. As far as phylogeography of H9N2 low path is concerned, there are again transmission links between East Asia, India, and Middle East. We also tried to study selection pressure, that which genes are under selection pressure. So hemagglutinin, that HA, NA, M2, and NS1, these are the proteins, and these are the sites which are under selection pressure, showing that ongoing evolution. As far as human cases are concerned, as we all know that first human cases were reported in Hong Kong in 1997. And as of today, there have been 882 cases with CFR, case fatality ratio, approximately to 50%. So as far as disease in humans is concerned, H5N1 or avian influenza viruses cause symptoms from mild flu-like symptoms or eye inflammation to severe acute respiratory disease and or death. Disease severity depends on the virus which is causing infection and the characteristics of the infected individual. So the first report, as Dr. Nivedita Madam mentioned, was in 2021 from an 11-year-old boy from Haryana who was admitted in AIIMS having underlying medical conditions, acute myeloid leukemia, and the boy had exposure to poultry and reported with SARI, severe acute respiratory illness, with multi-organ failure and succumbed to infection. This virus, which was found, belongs to the clade 2.3.2.1a. So as far as low pathogenic infections are concerned, this virus globally 115 human cases have been reported, and all of them were mild infections. As far as India is concerned, there was a report of H9N2 virus infection in a 17-month-old boy from Melghat district, Maharashtra, and severe pneumonia was reported, and the child recovered from the infection. So as far as we asked a question whether are there any crossovers, so we had performed earlier seroprevalence study in poultry worker, which is a risk group, in which we found antibodies against H9N2, but they were negative for presence of antibodies against H5N1 virus. So conclusions: outbreaks of HPI, H5N1, H5N8 have been reported in birds in India. These H5N1 viruses belong to 8 genotypes. H9N2 belong to 5 genotypes. Inter-subtype and interclade reassortments have been found. And H5N1 virus from human infection is having unique multiple reassortment and molecular markers for mammalian adaptation and high pathogenicity have been shown. So as far as crossing species barriers are concerned, avian virus, there are barriers. One is virus should cross species barrier. So from birds to humans, so these avian viruses are trying to jump into another species. Second is efficient replication should happen in human beings. And third is there shall be active transmission from one human to other human being. So here virus is crossing species barrier. It is replicating efficiently in human beings also. However, there are no persistent human to human infections. So this is something which is a good news. So where can we intervene? So way forward is multidisciplinary work with One Health approach involving human health, animal health, environment and forest departments to avoid another pandemic. So I thank all the group. I thank Dr. Rajiv Bail, sir, Dr. Sheila Godbode, Dr. Nivedita Gupta, Dr. Anoop, Dr. Jayati Mullik, students, Diksha, Sachin, Dr. Satish Pandey, long-time collaborator while working on wild birds and migratory birds, forest department, ICMR, ICMR, and all the collaborators. Thank you very much. Thank you.
Thank you very much, Dr. Shailesh, for that excellent overview of the various types and genotypes of avian influenza viruses. You also showed the outbreaks in India at different time points, also pointed out the seasonality and how the virus is evolving through reassortment and various other important details. Thank you very much for your excellent talk. Now we will move on to the next speaker, Dr. Farah Ishtiaq, who's a principal scientist from Tata Institute for Genetics and Society. Dr. Farah is an evolutionary ecologist who's interested in ecology and evolution of emerging infectious diseases. With expertise in avian ecology, she has held 2 postdoctoral positions and 2 independent fellowships following her PhD. She has been a Marie Curie Fellow at the University of Oxford and was also part of the Global Health Program Wildlife Conservation Society. In a multi-stakeholder partnership, she has established wastewater-based surveillance for SARS-CoV-2 and other infectious diseases in and around Bangalore city. So thank you very much, Dr. Farah Ishtiaq, for agreeing to deliver your talk. And over to you. We look forward to an interesting presentation from you.
Thank you so much, Dr. Nivedita, and good evening, everyone, and good morning if you're joining from somewhere else. I will share my screen. Okay, I think you can see my screen. Okay, so as you all just heard, is it all visible, my screen? Fine?
Visible. Can you put on full screen, please?
Okay. Is it now in full screen?
Not, yes, now it is, ma'am. Please carry on.
Okay, thank you. Right, as you just heard that I am actually a bird ecologist and now have been working on infectious disease for birds for the last 20 years or so. And avian influenza is something I have been very much interested. So I just would like to share some of my...
And we can see your duplicate screen.
Okay, wait. Let me just try it again. I'm not sure why is this happening. Is this better now?
Yes, ma'am. This you can put it in full screen mode.
Yeah.
Because presenter view doesn't work in Zoom.
Okay, is it fine now?
Yes.
Okay, thank you. Right, so birds are actually infected with, they carry lots of parasites, and they have been, a lot of bird populations are threatened with many parasites. So all you're seeing on the screen here is, on the left here is Trichomonas, which is a protozoan parasite which birds get infected with, and it kind of stops them from swallowing the food, and then they die. There's a huge population of green finches, which is threatened by this parasite in UK. But also you have something called West Nile virus, which was just mentioned. West Nile virus is through the mosquito, it's a vector-borne disease, and humans are dead-end hosts for this. I worked for a long time on avian malaria. It's a very different species of malarial parasite which infects birds, and a lot of bird populations are threatened with that. And then comes the avian pox, which again is a avian vector-borne disease. But I think from the One Health point of view, the concern really happens that when you have a parasite which can spill over or which can infect humans as well, or other species on this planet. And I think this is where this avian influenza or the viral diseases become very important. So as you just have heard that birds are a natural reservoir for avian influenza, for type A influenza viruses. And this subtypes can occur into any combination. There are at least 15 subtypes which have been found in birds, but only H5 and H7 are responsible for the full-blown outbreaks, which leads to the mortality in immunocompromised birds, but also mainly into the poultry sector. And then you also heard that it leads to, low path actually just leads to low egg production and everything. But if you look at the landscape, what we have in the Indian context, you do have different waterfowls or the wetlands, but also you have poultry sector, or say the pigs or other kind of livestock, which is kind of in the vicinity in the interface with all this environment. And this allows for a very good kind of assortment of this virus. And this antigen shift, when this happens, you see a different combinations of, say, H3 with N1 or H1 and N1, which is also known as swine flu. And these infections happen. And H1 and N1, which even though it evolved here, it also infects human now, which refer to as the influenza, which is a respiratory disease which comes and goes. But if you really look deep down into the ecology of the avian influenza, that why these outbreaks happen and why they have become so prevalent in the last few years in India, I think there is the answer is more complex than what we think here. So India has a very fantastic flyway. We are part of this Central Asian flyway just here in the green over here. And what that means is that a large number of migratory birds, they come and they winter here every year. Now, these migratory birds are not just ducks and geese or waterfowls, as we always think, that everybody think that's what migrant constitute, but you also get the woodland birds as well. But because the waterfowls or the wetland birds, they tend to live in big groups, or they huddle together, and the kind of habitat they share, the chances of any infection to transfer is, the chances are very higher. And most of the time, the species like, the ducks and the geese and the gulls, which are the natural reservoir, even if they're infected, it may not cause naturally mortality with them, but it will infect other species, which will show a sign of the disease in a different way. So it's a bit like in COVID that we are asked to do social distancing. I think in birds, if you have a segregation, I think that would work very well if they don't have to transfer this infection. So for a species infected with a subtype of avian influenza, they start excreting it as part of their fecal route. And they are around these wetland sites. When they are excreting it, the other species, when they come in contact with this, the fecal matter, or even with the water, they get also infected with. But also one of the reasons is that the virus, when the bird would stop excreting the virus, say, after a week or so of them in this infection, but they also, the virus can persist for even months in that, if the conditions are ideal or conducive for it to allow it to sustain. The map I'm showing you on the right side is from the Bombay Natural History Society, which highlights the important bird areas in India, and it actually flags most of the wetlands which we have, which are the large congregation sites, which are highlighted under Ramsar, convention that these should be protected for the wild waterfowl. Now, when you talk about avian influenza, I think the one country which really stands out for me and which really helps you to understand that how this ecology evolves is the Mongolia. I have done my postdoc here long time back, but I will just take a second to explain why it matters. So Mongolia doesn't have any poultry of its own. It's really minuscule amount, because you see the lot of population Mongolia lives is a nomadic population. I mean, they just move around every year. So there is hardly any poultry. So and it's a network of lakes where they have, these are salt lakes, which gets frozen in the winter. But Mongolia also have outbreaks of avian influenza, mostly in the breeding time, when the birds, they come from the China, from the north to in the summertime. But most of these species, when they start, they are huddling together and they are breeding in the big colonies. They're also shedding the virus, which infects other birds. But sometimes when you don't even have had any outbreaks, say in the north, then also you see sometimes some outbreaks happen in these localities. And that is a result of this virus, which has been kind of persist in these water within the low temperature, which is good enough to infect these birds. So these are the transmission routes which happen not just by birds coming in contact, but also fecal oral route as well. So as part of this, there's a lot of environmental surveillance, kind of passive surveillance, which has been done to understand that what are those species which are more vulnerable and which get has an impact of this disease. So there is a mute swan, as is showing you here. There's a shell duck, which also is a migrant species which comes to India. And on the spot, doing the necropsy and taking the sample and figuring out what kind of subtype the birds was, which is the H5 or H7, which was driving these outbreaks. And most of the time it was H5 at N1 outbreaks. Now, why this all matters in the context of India, that a lot of species from the north, they come to winter in India as well here. So this is a bar-headed grease. India gets the global population of this bird every year. And it's a very lovely migration route, which has been depicted here by the Bird Count India. And sometimes when these birds are also infected with influenza virus, they also bring it here. So you see sporadic chain of reaction when some of the birds come and they say mingle with other species in the wetland sites, and you see a chain of outbreaks. So this is a map from 2021. We had the very huge outbreak happen. I marked pretty much every bird which I found in the internet and also in the newspaper, which a species which was infected by this kind of outbreak. There was a large outbreak happen in these bar-headed geese, which is 5,000 birds found dead. This was in the Pong Dam. But as rightly just now, it was mentioned that crows, but also coots, but other ducks and geese. Now, one thing very interesting about these species these are not woodland birds. These are most of the birds which are associated with the wetland habitat, but also the birds which are scavengers, like the crows. They like and they eat the dead birds, as and when they find. So that was the cause. And it also triggers a chain reaction in the poultry sector, where you had culling happen in a lot of places. Now, the first time, as we just heard, the outbreak of avian influenza ever reported in India was in 2006, right? You don't hear these outbreaks happening. In fact, you can't even predict when this outbreak will happen. And yet, whenever it happens is kind of a mass scale. So why these outbreaks have become so dominant, so prominent in the last few years? And you still hear them somewhere in Europe, like the whole seabird colony in Scotland or in most of the England have gone because of these avian influenza outbreak, and mostly because of the gulls which carry the reservoir for this, and they are transmitting. So very nicely, every time I think the wild birds are implicated for these transmission every time when it happens, but actually that is not true. This is some of the data which is published some time back in this journal over here, which I'm giving the references here. So what you see that, yes, the outbreaks have happened in January to April, which is supposedly as called the migratory bird season, but also in the later part of the year in November, December. This is the time when the migrant birds come and they winter in India, and they kind of start migrating back by end of March. But what you also see interesting is that we have outbreaks in the non-seasonal time as well, which is in May, July, in August, September, and all of these are marked here on the map. And what it does tell you that despite not having these wild birds, you still have some kind of incipient kind of endemic transmission of these influenza viruses going on in the poultry sector, which being a notifiable disease, it gets later notified into the reported that there was an H5N1 outbreak. So it's not just the wild birds which is driving it, it's poultry itself is able to sustain some of these different subtypes which is driving this case. So now it asks the question that what could be the reason? And I think India has become a very fastest growing egg producer in the world. We have very large number of poultry farms. There are very lucrative, schemes which allow family to have these poultry farms in their backyard. However, I think the question remains that what kind of regulations are we following in order to make sure that poultry itself is safe and people are also safe, the kind of pollution and other, biological hazard which we are facing as a result of that. So just to give you an example, that a lot of wetland habitat has gone, so which also meant that with this shrinking waterfowl habitat, there is an increase in the connectivity between the wilder bird and poultry interactions, which leads to these are the drivers of the outbreaks, which leads to a lot of outbreaks both in the poultry as well as the wild bird. But you have a lot of poultry farms which are really just spread around, even in the urban areas, which some of them should not be there, very close to human vicinity. The lack of regulation, but also next to nothing, probably kind of monitoring in terms of very limited biosecurity. But as I just said, the density in the poultry farm, we know that viral diseases, they happen when everything is in very crowded space. And I would say we do have some surveillance, right? But it's kind of a post outbreak surveillance. And when the damage is done, then we start looking for what kind of subtype had caused that outbreak. And I would say what we need a system now is that where we need more. I mean, the only reason I think we controlled SARS-CoV-2 in the country that we had so many labs involved on the ground, looking for the virus and making sure that things get in control. And I think with few labs just looking for the wild birds or understanding this scenario, I think is not going to solve the problem. And this is where the environmental surveillance for the avian influenza can play a role, where you have, by designing these strategic protocols, and we build the capacity for this surveillance, and somehow we can develop an early warning system which can figure out what kind of low path and high path viruses we have floating in both in poultry but as well as in wild birds. So just to say a brief example, these are the sites which I have marked on this map. These are the sites which are monitored every year as part of the Asian wetland waterfowl census. This census happens every year. These are designated sites where a bunch of people who go and they document the wild birds in the January, February time, because that's the peak time when the wild birds are there, and they count and they report. This data gets collected and then it gets reported to Wetland International. They have these records for years, but the sites are there. So it would be, I think, very meaningful to map these sites over the poultry farms you have and figure out a system that can be have this certain selection of sites where such monitoring can take place, and we can agree on some kind of sample size and taking the soil or water samples and do some sampling strategy for pre and post season to understand that what kind of in avian influenza viruses are we seeing in this network. But also having kind of surveillance system where people integrate, as I said, that only few designated lab cannot solve the problem. So having more hands on the ground, I think an environmental surveillance can play a huge role. The reason we know so many SARS-CoV-2 variants are there in a community level today is because wastewater surveillance actually helps us to understand that. And I would say current measures are culling when the surveillance has happened, but I think we can really mitigate some of it, which I would love to hear from others, that what has been done, say, in terms of vaccination, because as far as I know, there is no currently vaccination for Indian population. We don't even know what subtypes are there, and most of these vaccinations are very subtype specific. And finally, just to give that what it means in terms of the One Health, that yes, death of the poultry has a negative impact because you have so many birds. It's a loss of socioeconomic loss, but it also is a kind of a zoonotic risk and economic loss from a human point of view. But also it also has a very negative impact of the emerging zoonosis, high disease risk. Most of the poultry farms are not very biosecurity controlled, so you do have pollution kind of pest, and it affects the ecology of the area. But having a surveillance program, I think, can really gear it up, not just for the poultry and humans, but also for the wild birds, because believe it or not, India attracts a lot of global population for so many species. And if this populations go, I think that would be a huge loss. Thank you. I'll be happy to take questions if there were any.
Thank you, Dr. Farah. That was an excellent talk. And you really alluded to some of the infections like avian malaria, West Nile, avian pox, which as we working on the human side, it is really, we were not aware of these infections. So thank you for enlightening us on that. You also spoke about the extent of spread of the disease, as well as how we have to be vigilant during the non-endemic season where the baseline transmission continues. And also, you pointed out on a very important gap that surveillance is weak. We mostly act after the outbreak has already happened, which results in culling of thousands of birds every year in various parts of the country, thousands or millions. So there is definitely a need to set up an early warning signal and a surveillance system, perhaps, which would be able to detect the infection preemptively and prevent the huge economic losses that we incur by culling of the birds. So thank you very much for your enlightening talk. And we would now move on to the next speaker, who is Dr. Timothy M. Uyeki, Chief Medical Officer of the Influenza Division at US CDC. His interests are primarily antiviral treatment and clinical management of influenza and respiratory viral infections, human infections with avian influenza A viruses, and other emerging viral infectious diseases. Dr. Uyeki is the clinical chair of the 2024 World Health Organization Influenza Clinical Management Guidelines Group and has been a member of the NIH COVID-19 Treatment Guideline Panel since March 2020. He is also a member of the WHO Guidelines Development Group on Therapeutics and COVID-19. He has been a consultant to WHO on seasonal avian and pandemic influenza, Ebola, SARS, MERS, and COVID-19. He's a clinical professor in the Department of Pediatrics, University of California, San Francisco School of Medicine. So we are delighted to have you, Dr. Uyeki, and over to you for your talk now.
Thank you very much. Can you see my slides?
Yes.
Okay, terrific. All right, thanks so much. I'm going to speak on some of the public health aspects of highly pathogenic avian influenza H5N1 virus. What I'll start out with is summarizing the epidemiology of human infections. I'll describe the public health perspectives from the US experience and our priorities. And then I'll sort of give an overview of our current assessment of H5N1 in terms of the public health threat and some of my thoughts on global public health priorities. So let me start with the epidemiology of H5N1 virus infections in people. So if we go back to 1997, and this was covered by earlier in this webinar, about the first case. It occurred in May 1997. It was a 3-year-old child. This child was hospitalized for multi-organ failure, sepsis, later found to have Reye syndrome, was a fatal case. And actually, it was not known what the etiology of this child's infection was until several months later, in August of that year, when the virus was isolated from a lower respiratory tract specimen and was found to be high-path H5N1 virus. This is the first time that we knew that a virus could go directly from poultry to infect a human and kill them. But it actually was not quite clear what the source of that child's infection was, although there were confirmed H5N1 outbreaks among poultry at Hong Kong poultry farms. And so we didn't hear too much more until 6 months later, when there were more human cases identified in Hong Kong. And overall, there were 18 cases with 6 deaths. Many of these cases occurred in children as well as young adults. The median age was 17 years, with a range from 1 year to 60. 11 cases had pneumonia, although 7 did have mild illness, and including many of the younger children. So this was a very important outbreak because, again, as I mentioned, it's the first time we knew a virus could go directly from poultry to a human and kill them. But we also had a wide range of infections here, from mild illness to very severe and fatal illness. Now, what Hong Kong did was they implemented a number of measures to try to contain that outbreak. They culled all poultry in the live poultry markets, about 1.6 million poultry. They stopped importing poultry from southern China. They cleaned up the markets. They disinfected the markets. They implemented a policy where you could not mix different poultry species. So you could not sell in the same stall chickens and ducks and quail and so forth. You had to segregate poultry species. And so that helped prevent transmission across species and reassortment. In addition, Hong Kong implemented a poultry vaccination strategy as well as China. And to date, Hong Kong uses it's actually a trivalent vaccine. It's 2 H5 antigens as well as H7. And the H7 is because of large outbreaks that started in Eastern China for H7N9 virus, which is a low pathogenic virus that then evolved into a high pathogenic virus. And there were epidemics of human cases from 2013 through 2017. So China uses a trivalent H5, H7 vaccine in commercial poultry. And anyways, Hong Kong implemented active surveillance for both imported and locally produced poultry. Now, there were a number of epidemiological studies, and we learned a lot from that outbreak in Hong Kong in 1997. So case control studies show that the main risk factor for humans to get infected with H5N1 virus was visiting a live poultry market in the 7 days prior to illness onset. There were other sero-epidemiological studies that were implemented, and some key findings were showing serologic evidence of transmission from patients to healthcare personnel. So there were 2 healthcare workers who seroconverted. They did not have any poultry exposure, so we believe that it was patient to healthcare worker transmission. One of them was asymptomatic, and so that is actually quite notable, and one had mild illness. In addition, there were other seroepidemiologic studies that showed that there were household contacts of some of these H5N1 patients prior to the patient getting hospitalized and isolated. So there might have been some limited human-to-human transmission that occurred. At least there was some serologic evidence suggesting that. And that 3% of the government poultry workers were found to have antibodies to H5N1 virus, but 10% of poultry workers. So it suggests there were additional human infections beyond the 18 that were identified with 6 deaths. We then didn't hear about human cases until February of 2003, when there was a family from Hong Kong that went into southern China to Fujian province for the Chinese New Year. And at that time, one of the children in the family died of pneumonia in southern China, was buried, not tested. The family returned to Hong Kong. The father and the son were hospitalized. The father died of respiratory failure. The son survived. So there were 2 confirmed cases, but then a probable case in the daughter who had never been tested, who died in southern China. So that's a family cluster, and that was pretty alarming. And if, for those of you who are working in clinical medicine and public health, back in early 2003, particularly February and March of 2003, and you started to hear about these reports of atypical pneumonia in southern China. And then in March and April of 2003, starting to hear about atypical pneumonia cases in Southeast Asia, and then cases in other regions of the world, including Europe, North America, et cetera. And we now know there was alarm at that time. There was concern about an H5N1 pandemic because of this family cluster. But now we know that it was actually the SARS epidemic, and that was SARS-CoV-1 that caused that. But that was something that, everyone in the world was worried about, an H5N1 pandemic. Anyways, as you've heard about the role of migratory birds spreading H5N1 viruses, I also believe that human beings are moving poultry across borders that can contribute to this. And the virus is endemic in a number of countries among poultry, so that we saw the emergence of human cases of H5N1 in different regions of the world, starting in Southeast Asia and East Asia, but spreading to other regions of the world, including the Middle East, Europe, and Africa. And so to date, as of today, there have been, since 1997, 907 confirmed H5N1 cases reported to the WHO, more than 50% mortality. It's about 52% case fatality proportion reported from 23 countries. Most of these cases had severe pneumonia. There have been very few cases reported, sporadic cases, since 2016. There have been mostly cases of avian to human transmission, but I'll mention some of the other cases. And most of these cases have occurred in young adults, but some cases have occurred in children. This is the epidemic curve of human cases worldwide since 1997. And what you can see is that cases have really dropped off since 2016. Big outbreak in Egypt was the last major outbreak. And you can see sort of sporadic cases since then. And this includes the Indian case that was mentioned back in 2021. So since 2022, there have been 24 cases reported from 8 countries. By my count, this is included 13 severe illness cases with pneumonia, including 6 deaths, mild illness in 3 cases, and asymptomatic infection in 8 cases. However, I do not believe all these cases were truly infected. There have been 8 cases, and that's in the red font here, including asymptomatic cases reported from the UK, 5 of them. 2 cases reported from Spain. These are all in poultry workers. And 1 case in the UK was an elderly man who raised ducks in England. These were all asymptomatic infected cases, or 1 case in the US only had fatigue. All these individuals had very, very low levels of viral RNA detected in a single upper respiratory tract specimen. None of them had, those that had serology performed, paired sera, they did not have evidence of seroconversion. I think what this represents is likely transient detection of non-replicating virus, not infection. And this has been reported in the literature. And so I think that truly probably been 16 cases with 6 deaths, but there have been 5 cases reported in 2024, all from Cambodia. It does include some severe cases, does include 1 case of asymptomatic infection, but also some mild cases. So the cases in Cambodia are of different clade than all the other virus infections. Worldwide, we're seeing that the clade 2.3.4.4b H5N1 viruses are widely circulating among wild birds. They're spilling over to cause poultry outbreaks and also infections in terrestrial mammals, but also some marine mammals. You've probably heard large outbreaks in seals, elephant seals, and so forth. So this virus has spread among wild birds to other wild bird species, and some spillover to mammals in many regions of the world. And there have been some associated poultry outbreaks. So we saw last year the first human cases in South America. It's the first time we've had high-path H5N1 virus in South America in wild birds and poultry as well. And so what are the risk factors for H5N1 virus infection? Generally, this is direct or close unprotected exposure to sick or dead infected poultry or raising backyard poultry. The virus is in the respiratory and gastrointestinal tracts. It's shed in the feces. The virus can be aerosolized when one is handling birds or when birds are flapping their feathers. And then one can inhale virus into the lower respiratory tract or the upper respiratory tract. Also, a risk factor we know from a number of studies is visiting a live poultry market where there were infected birds. So you don't actually have to have direct or very close contact with birds, but if virus is aerosolized and the virus is inhaled, then you could get infected that way. There have been 2 clusters way back in 2006 in Azerbaijan, where there were people who were defeathering wild swans that had died of H5N1 virus. Probably the virus was aerosolized virus was inhaled. There were deaths in these cases. There have been a small number of cases with an unknown source of infection. We did have one imported case and a return traveler to Canada and North America. That patient died of pneumonia and meningoencephalitis. And there are some cases in other countries where it's not quite clear what the source of infection was. There have never been a case of mammal to human transmission, although there have been many wild mammals as well as some domestic mammals, domestic pets as well, that have been infected with H5N1 virus. We have seen some clusters of H5N1 virus, mostly in family members that epidemiologically linked by location and time. Most of these cases had common exposures to poultry, and they represent poultry to human transmission. However, there have been a small number of case clusters in which limited non-sustained human-to-human transmission likely occurred. These are secondary cases who did not have poultry exposures, but they cared for a sick H5N1 patient. This is as a result of having prolonged, unprotected, very close contact with a sick individual. This has occurred in households as well as in hospital rooms. And this has been documented in a number of countries, including second-generation human-to-human transmission in Thailand and China, but also third-generation transmission, so chicken to human to human to human. Dead-end transmission, this has occurred in Indonesia and Pakistan. And we have had, all of those were blood-related family members, so that's a parent and a child or siblings. But there has been patient to healthcare worker transmission, so non-blood related individuals in Vietnam. And I'll just say there have been other avian viruses, including H7N9 virus, in which there have been more cases of non-blood, of human to human transmission, non-blood related. So we've seen non-sustained, limited human to human transmission, but dead-end transmission. The virus has not had the ability for sustained human to human transmission. The clinical presentation, the incubation period following poultry exposure to symptom onset is typically 3 days with a range of 2 to 7 days. And most people have influenza-like illness, upper respiratory tract illness, signs and symptoms with or without fever. Most patients do have fever, but they can also have gastrointestinal symptoms such as vomiting and diarrhea. For those that do progress to lower respiratory tract disease, this tends to occur around days 4, 5, or 6 after symptom onset. There is progression to signs of lower respiratory tract disease, such as difficulty breathing, shortness of breath, chest pain, tachypnea. And at hospital admission, which occurs typically around day 5, 6, 7, patients present with signs of pneumonia, hypoxia, and there are some key laboratory findings, but they're not typical with every patient, such as low white blood count, low lymphocyte count, and mild to moderate platelet count. Signs and symptoms are accompanied by pneumonia. This is on the left, some cases of pneumonia in the outbreak in Vietnam back in early 2004. And then on the right, some cases, these are radiographs I took of cases in pneumonia, confirmed cases, a fatal case in the top, and actually a case that survived below. It's typical bilateral pneumonia. So let me move to the US experience and some of our priorities. So we have established H5N1 testing capacity at all state health department virology labs. If we do have a case that tests positive for H5N1 at a state health department, the specimen is sent to US CDC for confirmation. In collaboration with state and local health departments, we've been monitoring people who are exposed to H5N1 virus infected birds and other animals. And we have been analyzing H5N1 viruses detected in birds and mammals worldwide, as well as in people worldwide. We want to characterize those viruses. If we do have actually virus isolates, we will characterize them antigenically, we'll sequence them, we'll do phylogenetic characterization. If we don't have the virus, we'll look at the genetic sequences that are available online. if we have viruses, we are testing them both for genotypic markers of susceptibility. We're analyzing them, but we will do phenotypic assays to look at in vitro susceptibility to approved antivirals. And we're always working on candidate H5N1 vaccine virus development for the WHO. We have made recommendations for monitoring exposed persons, testing of exposed persons. We have infection prevention and control recommendations for suspected and confirmed cases. And we do have recommendations for the use of antivirals for both treatment as well as post-exposure prophylaxis. We collaborate with our agricultural partners, particularly in analyzing H5N1 viruses collected from poultry and other animals in the US. And we have assisted with surveillance worldwide, as well as case investigations of H5N1 cases in other countries, includes technical assistance to Cambodia to all of their cases identified last year, as well as this year. We have confirmed H5N1 virus infections of humans in the 2 cases in South America last year in Chile and Ecuador, as well as all of the Cambodia cases. Now, to date, since 2022, in the US, we have detected more than 8,500 detections in wild birds in 50 US states or territories. It's actually 49 states and the District of Columbia. There have been more than 1,000 outbreaks in commercial poultry or backyard flocks, totaling almost 82 million birds in 47 states. There have been 200 detections in mammals, 19 different species in 26 states. We have monitored almost 8,000 people exposed to infected birds and poultry in 52 jurisdictions. 186 of these people who were monitored became sick and were tested for influenza. We've only had 1 person that tested positive for H5N1 virus. That was in a person who was culling poultry, who was using PPE, but was not compliant with all of their respiratory protection. That person only reported fatigue. In a single upper respiratory tract specimen, they had a very, very low level of viral RNA detected. In a second specimen was negative. I don't personally believe that person was actually infected, but because we did detect H5N1 viral RNA in a human respiratory specimen, it did meet criteria for reporting to the WHO. We did report that case, but again, I don't believe that patient was actually infected. So let me move to our current assessment and global public health priorities. So right now, we believe that the current global public health threat of H5N1 viruses is low, but clade 2.3.4.4b H5N1 viruses are continuing to spread very widely among wild birds with spillover to poultry and mammals. This virus might get entrenched in poultry. It's clearly spilling over to cause poultry outbreaks and also outbreaks in mammals. It's not completely clear that there's mammal-to-mammal sustained transmission going on. There's probably been some mammal-to-mammal transmission, certainly in farmed ferrets, farmed mink, some wild marine mammal outbreaks, but it's not clear that this virus is actually established in any mammalian reservoir. It's pretty clearly established in wild birds and very efficiently transmitted, but we don't believe these viruses are transmitting very efficiently to people. And these viruses currently lack tropism for the human upper respiratory tract. I think if that were to happen, then the public health threat would be greatly increased. Now, there's also other clades of H5N1 viruses, particularly the clade 2.3.2.1c viruses that are enzootic. They're endemic among backyard poultry in the Mekong Delta throughout Cambodia, but they're also in Laos and Vietnam, and there have been these sporadic cases. So also, there's no evidence of any human-to-human transmission of H5N1 viruses since 2007. We're always worried about that, and because you've heard about the potential for reassortment, and these viruses clearly have reassorted, in some avian species. We believe that these sporadic human infections are going to continue to occur in exposed persons, particularly in these countries with endemic spread among poultry in rural areas. It's very difficult to control backyard poultry with these viruses infections. Now, what are the global health priorities? Well, first of all, I think every human case of H5N1 virus infection needs to be fully investigated, because the key question to answer is there human-to-human transmission that has occurred. And we need to determine if these viruses have the ability for increased transmission to and among people. So clearly you've heard that the key is global surveillance among birds. We also need better global surveillance among people at the human-animal interface. We need clearly to track the evolution and spread of these viruses. We need better collaborations among animal health and human health. We need better sharing of information, sharing of viruses rapidly. We need to work together to prevent and control these outbreaks in poultry because of the threat to human public health. We need better diagnostic capacity to detect these viruses in public health laboratories worldwide. And I think we need to strengthen infection prevention and control of acute respiratory illness in healthcare facilities worldwide. Even though we had a COVID-19 pandemic, we have an ongoing threat of respiratory viruses in humans. We need to improve public health measures to prevent the spread of human respiratory viruses in healthcare facilities. We need to improve our clinical management of seasonal influenza, and we need to expand influenza vaccine production and utilization in people worldwide. So let me just close by reminding us of the threat of other viruses. So you have heard about from some of the other speakers today about other avian influenza viruses. It's not just H5N1 viruses. And so I'll just say that this slide shows a table of low pathogenic virus subtypes and other highly pathogenic virus subtypes that have transmitted from birds to people to cause either mild disease, which I call uncomplicated, or severe disease, as well as fatal disease. So it's not just H5N1 viruses that are the threat to human public health. And don't forget variant or swine origin influenza A virus infections. This whole webinar is focused on avian influenza and the threat, but don't forget swine origin influenza A virus infections. And why do I say that? Because what are we worried about? Yes, we're worried about sporadic human infections or zoonotic influenza, which can be from avian or swine influenza viruses. But what we're really worried about is the next human pandemic, influenza pandemic. And we know from experience, pandemics are rare, but when they emerge because of a novel influenza A virus has come out of an animal reservoir. And there has been reassortment. Either we know historically this has come from an avian reservoir or a swine reservoir. And I don't think I need to remind you that the last influenza pandemic in 2009 was from a swine reservoir, and it was a reassortant virus with genes from birds, humans, and pigs. And so, yes, it emerged out of a swine reservoir, but it became a human virus. It was no longer a swine virus. And so the ecology of influenza A virus is on the left in this blue square, just is a reminder that influenza A viruses circulate among many animals, not just among birds, and are a threat to humans. And so I just want to close by saying, yes, all avian influenza viruses that have infected humans pose pandemic potential, but so do those from swine origin viruses. And there's a wide range of clinical severity from asymptomatic to mild to very severe disease in infected people. And so we need to improve our response to seasonal influenza, and that will, I think, prepare us better for the next influenza pandemic. So thanks so much. And then on our CDC web pages. We have many, many resources available for those of you in public health and clinical medicine, many recommendations, and I'll stop there. Thanks so much.
Thank you, Dr. Uyeki, for the excellent talk and enlightening us on many, many issues related to the epidemiology, the risk factors for acquisition of infection, then also the alarming trends of human-to-human transmission, both second as well as third generation transmissions you discussed. But the heartening thing was that still it is a non-sustained transmission. And you said that the public health threat at present is low, but definitely we need to be vigilant for any evolution of the virus further and it getting the capacity to establish human-to-human transmission. You also enlightened us on the surveillance system that has been established in the US, where you are also looking into the various vaccines and antivirals, as well as setting up a public health surveillance system. And also very importantly, pointing out that we should not forget to look at the non-avian reservoirs of these viruses, influenza viruses. So thank you very much for that very enlightening talk. I would now like to invite the next speaker, Dr. Chakradhar Tosh, who's a principal scientist at the ICAR, National Institute of High Security Animal Diseases. Dr. Tosh has been working on animal health for close to 30 years in different scientific capacities. His body of research work includes genetic classification and pathotyping of influenza viruses, particularly avian influenza viruses in India. He has isolated several avian influenza viruses also. He has developed NP gene-based RT-PCR assay for diagnosis of avian influenza and identified small inferring RNAs that inhibit in vitro replication of AI viruses. Currently, his research work focuses on avian influenza diagnostics, molecular epidemiology, and vaccines. Over to you, Dr. Tosh, and we look forward for a very exciting talk.
Good evening. Thank you, madam, for this opportunity. I am audible.
Yes, you are.
Yeah. Thank you. And you heard about the avian influenza affecting poultry, wild birds, and humans, and also it affects the livelihood in poor countries and also government spending to control this disease. And besides avian influenza, we also work on the exotic and emerging animal diseases from animals. And this is one of the outbreaks. These dogs is local breeds in Kerala was affected with the hypath H5N1 avian influenza. As you know, these are some of the recent outbreaks reported during 2022 and 23, and huge mortalities were reported in free-range dogs in Kerala during October 2022, and H5N1 virus was detected. And these are the works carried out at NISA at Bhopal, and this outbreak was continued in backyard farms and farmed poultry till January 2023. And these are the birds involved in Kerala outbreaks: chickens, dogs, and quails. All the viruses were H5N1 subtypes. Then during same time period also, there are outbreaks in Jharkhand in the chicken farms, and also in Bihar, also chicken farms, all are H5N1. During September 2023, we isolated viruses from a wild bird, that is black swan, and a silver pigeon from the Mumbai, Maharashtra, from a natural park. And these viruses were sequenced, and they are characterized here at National Institute of High Security Animal Diseases, Bhopal.
Dr. Tosh, can you do the full screen here? Presentation mode, please. Thank you.
Is it now full screen? Okay.
Now it's better, sir. Thank you.
Yeah. Then during same time period, we have also outbreaks in the neighboring countries like Bhutan and Nepal, they are all from domestic birds and also wild birds, including crows, and all viruses were H5N1. China also reported during same time wild birds from bow-headed gull, and that is belongs to the clade 2344b, which is currently circulating across Eurasia, Africa, and Americas. All these viruses in 2022 and 23, they are sequenced, and phylogenetically they belongs to two major clades, that is 2344b and another is 2321a. This 2344b is related to the virus. The Indian viruses are related to the viruses circulating poultry and wild birds in Eurasia, Africa, and also Americas. And since based on this genetic characterization and the relationship and the time of outbreaks, this outbreak, the source of the virus is predicted to be the wild bird migration in the Central Asian flyways. But however, this 2321A that shares genetic relationship with H5N1 viruses circulating quite some time in SAARC subregions, including Bangladesh, Nepal, and this could be one of the regions of environmental persistence or informal trade, because similar viruses are circulating in the neighboring countries at the same time period. So these two regions could be one of the informal trades, because these borders in the SAARC countries or the South Asia, they are very porous, and there may be movement of birds across the borders. So that could be one of the cause, or there may be environmental persistence of the virus 2321A, which is causing the outbreaks. These are some of the influenza, avian influenza H5N1 outbreaks, viruses infecting humans. And I think that is, I just want to correct, because from the US CDC, it is from 23 countries, 907 human infections, and including the Bangladesh, China, India, Myanmar, Nepal, and Pakistan. These are the outbreaks reported that indicates the zoonotic importance of this virus. It has already been discussed. What are the risk factors of this virus, high-path AI into India? There are some of the regions, the wild bird movement. As you know, this is the Central Asian flyway that spans between the Arctic region to Indian Ocean. And there are millions of birds migrate twice a year during north and south directions. In October and November, they come back to this India and then go back by February and March. There are a lot of wetlands these wild birds visit. There are around 75 Ramsar sites or wetlands in 23 states in UTs, and these wild birds stay in these areas for 4 to 5 months and then go back to their own place, like Siberia or the Arctic region. And there is another risk factor is huge poultry. Indian poultry is more than 850 million poultry, with 40% around backyard poultry, and there is very low or no biosecurity issues in the backyard poultry. So that could be one of the causes causing outbreaks and sustenance of the virus in India. Then there is live bird, because in India we prefer the fresh dressed poultry. So live bird markets, and there is market chains also there, and there is very less biosecurity issues. So the virus may persist, and there is chance of interaction with humans and human-animal interaction, and there is chance of transmission or the spillover of the virus to humans. And there is another domestic ducks that plays an important role in they act as a bridge species between wild birds and also domestic chickens, because they don't have much clinical signs like chickens, but they share virus up to 17 days. So they could be acting as a bridge species between the wild birds visiting the wetlands and between the chicken farms. And there is a huge international border, more than 15,000 kilometers in the SAARC countries. And these borders are mostly porous, and that encourages some informal trades and movement of poultry along with infected birds. These are some of the outbreaks since 2005. The virus was detected in Qinghai Lake in China, and then this virus spread to other regions through bird migrations. And this virus, this region, South Asia is the most affected by H5N1 or H5N8 viruses in poultry. More than 14,000 high-path AI outbreaks has been reported in 6 countries, including Afghanistan, Bangladesh, Bhutan, India, Nepal, and Pakistan since 2006 till 2023. There is an emergence of multiple genetic clades already cited by previous speakers. Like earlier, these genetic clades were 2.2, then till 2010. From 2011, there is emergence of a 2321A virus, which is still circulating in South Asian regions. And then there is emergence of 2321C, which was very briefly in 2014 and 15 in 3 states. Then came the H5N8 virus of clade 2344B, and now the H5N1 virus with 2444B. So these clades are circulating, but currently the major clades are 2344B and 2321 viruses. The earlier clades, they have disappeared, but these are the major clades now circulating in SAARC countries. These are some of the outbreaks since 2006 till date. There are more than 350 outbreaks has been reported from 24 states and union territories. And H5N1, H5N8, and another one is human infection in 2021 was reported. And this virus has been detected through surveillance from the domestic poultry, domestic birds, including crows. There are a lot of crow mortalities since 2008, and huge outbreaks reported from 2011 till date. Virus also isolated from wild birds, environmental samples, and live bird markets and humans. And the outbreaks, if you see the period of outbreaks, the outbreak starts from October until April, and the peak activity of H5N1 was observed in January. And since this South Asia is dense human and poultry population, there is human-animal interaction in the live poultry markets, and there is chances of spillover of the virus to humans, and emergence of a pandemic strains may likely happen in. So there is a need for preparedness for this, and these are some of the reports for bird migration that is bringing the virus or spreading the virus in South Asia, including India. These are the outbreaks analysis from 2008 in 2010. There are 3 times there is introduction of the virus to the Indian poultry population. Then in 2014 and 15, there is a new clade emerged in 2321C, and that virus, based on the median joining network analysis, it was found to be spread through wild birds and linked to a wild bird in Middle East. Similarly, the H5N8 2344B clade, there was huge outbreaks in 7 states and during 2016 and 17, and a new virus that is not reported earlier was found, that is H5N8 with 2344B. Based on the time of outbreaks and the genetic relationship and the dispersal of the virus, this virus was found to be wild bird origin, and then internal movement was through maybe poultry or wild birds. Of course, only wild birds we cannot blame. There may be informal trades between the countries, because we see that there are a lot of viruses circulating across the borders, and the porosity of the borders in South Asia. So that indicates the informal trade between the countries may be one of the reasons for spread of the viruses in the South Asian region. For the vaccine development as a preparedness, we see that there are major clades 2321A, 2321C, and 2.2. So we wanted to see how this virus as a preparedness for future vaccine. We see how this, if one vaccine developed with one clade, whether it is giving antigenic coverage to other clades, other viruses or not. And we see that there is a lot of, besides with genetic divergence, there is also divergence antigenically in these clades. So a vaccine with one clade may not give protection, because the viruses are moving across the called territories through wild birds. So if first vaccine strain is developed with one clade, that may not give protection to other clade viruses circulating, coming in the next, emerging in the next seasons. So what are the control and containment measures taken by the government of India? This is the action plan. We already have the Department of Animal Husbandry and Dairy, and test and slaughter policy is the choice. There is no vaccination policy for high-path AI, and after the slaughter policy, there is sanitation and sanitary certificates is issued by the state governments. And then there is compensation of the farmers for the forced culling of their birds. Then after this compensation and sanitary certificates, then there is post-operative surveillance plans, which continues for 4 fortnights, that is around 2 to 3 months, collecting out samples from 1 kilometer to 10 kilometer radius area, and then testing. And then if all the 4 POSP samples are negative, then the country is declared freedom from high-path. And there is also information and education and communication campaigns and there is Avian Influenza advisory and biosecurity and hygiene measures have been given. And also there is guidelines for zoological parks. If there is any outbreaks reported in birds or wild birds, then there is a guideline for how to handle this outbreaks in zoological parks. And also there is a compartment, even in the outbreak times also, the trade can continue from these compartments. And there is also surveillance activity is mentioned in this poultry and wild birds. Both the active and passive surveillance is continuous activity. And the district epidemiological veterinary officer is the nodal officer to collect samples from the identified areas as in the action plan, and that samples are referred to the regional disease diagnostic laboratory spread across the country. There are 6 regional disease diagnostic laboratories and 1 central disease diagnostic laboratory. And these samples, if they are tested positive for influenza A virus, then they are referred to the central laboratory at NISA Bhopal for confirmation, and then reporting to the Department of Animal Husbandry for reporting and then action plans. Then besides that action also, we import a lot of live birds, the livestock and the livestock products, and these samples are tested. These samples are referred by the animal quarantine and certification services. There are stations at New Delhi, Chennai, Kolkata, Mumbai, Bangalore, and Hyderabad. Whatever the imported the livestock and livestock products are imported to India, they are tested before release to the India. So these are the control measures followed by the government of India for control and containment of avian influenza. And there is another zoonotic influenza pathogen of low pathogenic avian influenza has been identified, and since 2000. Low path H9N2 avian influenza virus, and already it has been mentioned this virus is also affecting humans, including India. It has been reported, and this virus also contributing, the H9N2 low path AI also contributing to the emergence of influenza viruses which are lethal to humans like H5N1, H7N9, H3N8, and H10N8. And all the viruses, whatever isolated in India, they belongs to G1 lineage based on the hemagglutinin gene of the virus. And
Dr. Tosh, we have lost you for a moment.
The poultry industry and has been selected. The vaccine strain has been selected based on the hemagglutinin phylogeny, what is circulating in Indian subcontinent, and then antigenic cartography and growth kinetics. This has been commercialized. It will come very soon to the market for vaccination of the poultry for controlling the H9N2 low pathogenic avian influenza. And there, as a preparedness, we have also developed at NISA the reverse genetics based H5N2 vaccine. We're using the H5N1 viruses circulating in India and H9N2 viruses. And this virus, high-path virus, has been mutated to become a low-pathogenic avian influenza. And this is giving good protection till 200 days with high serum antibody titers. This is as a preparedness. And for pandemic preparedness, the information sharing of the situation in the country is very important. Once this virus is tested and detected positive, they are immediately notified to the OA, that is World Organization for Animal Health, since India is one of the members of this organization. And then this sequence information is also shared with WHO twice a year. These are coordinated by the OPFU, that is OA and FAO network of influenza experts. So twice a year we share the animal influenza sequences from the India or the neighboring countries whenever the samples are referred to NISA for confirmation to WHO for pandemic preparedness. And we also share the information with the Asia Pacific Regional Network of Experts on Avian Influenza Forum. And this information is also shared with the Ministry of Health and Family Welfare when there is a report of outbreaks. And this, as a pandemic preparedness, the veterinary services of Government of India has been evaluated by performance of the veterinary services by OA, World Organization for Animal Health, and the strength and weaknesses has been identified and recommendations has been given that will be beneficial for the veterinary services to come into the mark. Then there is a development of pandemic preparedness plan for respiratory viruses in India, and the plan has been developed already very recently. And there is also animal pandemic preparedness initiatives and World Bank supported animal health support for One Health. And this is the comprehensive plan to address the animal pandemics in India. To conclude my topic, it is already known that the virus is expanding its host range and also geographical range, except I think Australia and to the Antarctica. The virus already it is in the Eurasia, it is Africa or the Americas. So this virus is very difficult to eradicate in near future. So to control this disease, strengthening the intersectoral coordination for sustained One Health approach to control avian influenza is important as it is everyone's health. And that's from my side. And thank you for watching and hearing. Over to you.
Thank you, Dr. Tosh, for that very enlightening talk. You described the molecular phylogeny, genetic relationships between different types of avian influenza viruses, the emerging clades and subclades, and the role of wild birds predominantly in spreading of the infection across the borders. You also talked about or alluded to factors which contribute to the spread of viruses in the Indian continent, and what are the measures for control and containment. And also you alluded to the much needed vaccine R&D, which everybody is looking forward to. So thank you very much for that very comprehensive and enlightening talk. We would now move to the session on moderated discussions. And I would ask some of the questions which have been preemptively sent to us by the audience. And I would be directing these questions to the specific speakers. So first question goes to Dr. Sindura, where it has been asked that, is there a common portal which links human and animal health data? Would you answer that, Dr. Sindura?
Yeah, so currently, when we talk about data, we are talking about multiple types of data, whether it is clinical data, whether it is surveillance, active or passive, from which type of source it is, whether it is laboratory data, and so on. So right now we are in the process. The short answer to your question is we don't yet have it, but that is one of the goal of One Health mission, and we are working through that process and what is the best way to do it. without really compromising the work that is happening within the individual ministries or areas. And the idea is to have a federated approach where data can live in its wherever that is currently being housed, whether it is currently IDSP in the human surveillance, and in future, whatever evolution it does because of Ayushman Bharat digital mission. And in the animal husbandry, they are rolling out National Digital Livestock Mission, which will integrate all of the animal husbandry data. And wildlife, currently there isn't an integrated database itself. So that is being designed as we speak under the National Referral Center for Wildlife. It's a system across the country for managing wildlife data. So as you can imagine, it's a fairly uphill task of ensuring that even within each of these sectors, there is a linked and usable system in place, information system in place. And then comes how you link each of these. So it is currently in the works. We don't yet have a short answer. Short answer is no, but it is being worked out. Thank you.
Thank you, Dr. Sindura. The other question I would direct to Dr. Shailesh Pawar. And please feel free to refer the question to some other speaker if you are not comfortable in answering. I just did it as per my wisdom. So, Dr. Shailesh, the question is, what measures can be taken to prevent avian influenza?
Yeah, so measures means one of the measures could be avian influenza surveillance in wild and migratory birds. I mean, and then another is poultry surveillance is already in place, clinical surveillance is in place. So surveillance is the key so as to keep watch on circulating avian influenza viruses. So that is one of the important measures. And then also some of the other countries are using vaccines so as to prevent avian influenza. But as Dr. Tosh has mentioned rightly, that at this point in time, there are no vaccines against HPI, LPI used in our country. So that is another measure so as to prevent avian influenza. Thank you.
Thank you, Dr. Shailesh. The next question I would like to divert to Dr. Farah Ishtiaq. It's like can pet birds cause avian influenza?
No, I mean, I think the answer for that would be, for me, no. I mean, unless somebody is infected with avian influenza themselves and they transfer it to their pet, like it has seen in hamster and all, people get COVID and they transfer it to their pet. I think that kind of transmission can happen, but it's never been reported like that. I mean, influenza, as we know, I mean, avian influenza is usually harbored by in large congregations, you know, where you have conditions are very good and people who come in contact with someone who's already infected with this. So yeah, that would be my short answer for that.
Thank you. And my next question is for Dr. Uyeki. What are the thrust areas of research in avian influenza?
Sorry, could you repeat that question?
Yeah, I would repeat it. So the question asked is, what are the thrust areas for research on avian influenza?
Yeah, I mean, there are huge number of gaps. I mean, there are many, many areas of research. And so, you know, I don't think I can comment or should comment on the sort of the avian side because I focus really, you know, I'm a clinician and epidemiologist focusing on the human side. I would say from the human side, what we really don't understand is why there are so many people that have been exposed to different avian influenza A viruses, but a relatively very small number of people who are actually infected. Although I think we're under detecting, you know, a lot of infections. We're focused on severe disease. That's where surveillance is really on hospitalized people with pneumonia. We're not really looking so much for mild illness. We don't quite understand why poultry workers, you know, who are the most exposed worldwide, we don't seem to see a lot of cases in poultry workers. We see a small number, and we do know there's serologic evidence, but we don't see lots and lots of infections. So the question is, why is that? Do they have some kind of immunity from exposure to different avian viruses over time? And then we don't truly understand what is going to increase the risk of human-to-human transmission. We believe that the viruses need to be able to infect the human upper respiratory tract to be able to spread more among people, but we don't know. And so, I mean, I think there's so many unanswered questions, particularly in the immunology, susceptibility of why and how people actually get infected. If you look at what's happening in Cambodia this year, there's probably many, many, many people that have been exposed to sick or dead poultry, and yet a very, very small number of people that have been identified who have been sick. Probably a few cases have been missed, but we're not missing millions or hundreds of thousands of cases. So I think there's just a wide number of unanswered questions. And then from the clinical management side, we don't really have good treatment. We recommend antiviral treatment, and I would recommend it to any hospitalized patient or even patient who's not hospitalized with H5N1 virus infection or other avian influenza virus infections. But we actually don't have good treatment, which is basically supportive care. And so I think we need to be able to manage severe influenza, both seasonal influenza and zoonotic influenza better. Just to say there's so many unanswered questions. Thank you.
Thank you, Dr. Yoki. Now, my next question goes to Dr. Tosh, that what are we doing for developing a H5N1 avian influenza vaccine, and where are we on that? And this is for poultry.
Yes, as already showed, we have DISAD has developed vaccines for H5N1, that is reverse genetics vaccine, that is H5N2. N2 we have incorporated to see whether the poultry are vaccinated or they are infected, because when there is H5N1 infection, so we can differentiate whether these viruses are vaccinated, these birds are vaccinated or viruses. But there is no policy, but we have developed a vaccine, reverse genetic vaccine. Only we can insert whatever these strands circulating, that can be inserted into the reverse genetic system. But there is no policy so far for the vaccination because of obvious reasons, and we see the vaccination countries, the experience of the vaccinating countries. And initially we have India has stockpiled vaccines for H5N1, and when there is a need for vaccination, it was for the containing the disease. But now this stockpiling has been stopped, and now it is we have developed the vaccine. But if you see the experience of the vaccinating countries like China, Bangladesh, Indonesia, Egypt, or Vietnam, the vaccination is not the only choice. It is one of the comprehensive, items in comprehensive control program, including surveillance. Surveillance is a major issue. Surveillance is always a major issue. Surveillance, and then the biosafety, major biosecurity and biosafety issues, and then calling the birds infected. Even if you vaccinate, it does not stop the spreading of the virus. It only stops the clinical signs. It does not stop the spread of the virus. And there is issue of complacency and the coverage of the vaccine. If you don't cover with 60 to 80% poultry population, then there is no use of vaccinating the poultry. So there are so many, and there is a issue of cold chain maintenance, and what is exactly the virus circulating? Then that virus, that is the vaccine strand should give the coverage to the circulating strain. So there are major issues, but yes, we have already developed a vaccine against H5N1. There is a platform. We can insert any virus comes, whether it cleared, then the HA gene can be replaced, and it can be used as a vaccine.
Thank you, Dr. Tosh. The next question is to Dr. Sindura, that how this One Health can be implemented in tribal areas where multiple basic challenges people suffer from multiple basic challenges?
Yeah, that's a difficult question and important question, because we in tribal areas, the issue of zoonotic diseases is just one part of it. And more studies are showing how in India, it's quite a high prevalence of genetic disorders, whether it is sickle cell or other such genetic disorders in quite high numbers. And there is also quite high number of non-communicable diseases, whether it is typhoid or cardiovascular disease or diabetes and things like that. And you have to place One Health type issues, whether it is close proximity to animals and wildlife species, how much of that is an issue in the health system. So I think it all comes down to, when it comes to handling zoonotic diseases and these One Health diseases, there is a set of basic premises that we need to put forward, whether it is in tribal or rural or urban areas. There are slight variations to it. That involves, how do you link information from clinical aspects? How do you put in surveillance in place, whether it is both routine surveillance, and how do you make environmental surveillance into this, and how do you build action to that? So that's a separate category. Specifically for tribal areas, I think it comes down to having a robust health system. If we don't have health systems that enable care for them, it would be very difficult to think about other things, because tribal population doesn't just show up with one thing. They will show up with multiple issues, including basic clinical care. How do you have pregnant women having access to delivery service? Very basic, nothing to do with diseases. So I think we have to go back to basics to answer your question. That includes having a proper access to care overall. And on top of that, we build some specific issues that may emerge in certain areas. Brucellosis may be a particular problem where goat rearing is very common, or scrub typhus or some other such diseases in other areas, depending on the vector population. So I think if focused effort would be needed. What you're pointing out is right. Focused effort would be needed to address the needs of tribal population when it comes to this kind of issues, on top of the routine care that we have to provide. I'll just say that it's not a very satisfactory answer, but it's a fairly open-ended, broad question.
Thank you, Dr. Sindura. Dr. Shailesh, the next question is to you. That is there any surveillance for antibody-based surveillance for high pathogenic and low pathogenic avian influenza viruses in India?
Yeah, I mean, the question is whether it is in human beings or birds is not very clear, but definitely for birds, there is no serosurveillance as of now. As far as humans are concerned, there were some studies carried out. And as I showed, in one study, we showed antibodies, but again, to a very limited extent, around 5% poultry workers had antibodies against H9N2. But there were some serosurveillance, there are some serosurveillance reports in humans, but regularly there are no surveillance being carried in our country. In birds, maybe Dr. Tosh can give more information about seroprevalence in birds. Thank you.
Thank you.
Yes,
So,
there is a serosurveillance in the action plan of Government of India. And as far as H5 is concerned, there is no positivity, because the birds die very within 48 to 70, and serum development takes immunity maybe at least 5 to 7 days. So for H5N1, we have not seen any positivity of birds having seropositivity for H5N1. Yes, but H9N2, we have a lot of seropositivity, and besides that also we found as a differential diagnosis Newcastle disease virus seropositivity. So there is H9N2 seropositivity in poultry farms. We have seen already in the poultries, in chickens and dogs also. Thank you.
thank you. Dr. Farah, the next question is to you, that whether there is any alternative to culling when we detect an avian influenza outbreak?
I think culling is the last resort, right? When the outbreak has already happened, the alternative is there to strengthen the surveillance, really. So, and I think with respect to avian influenza, which is so different from SARS-CoV-2, there is no seasonality for SARS-CoV-2 so far. You have different waves coming in different time of year, but as a lot of data shows that everybody is implicating, wild birds are coming and there are outbreaks happening in different parts of the world. I think a lot of this predictive modeling can be done beforehand and surveillance can be strengthened, which should be carried out throughout the year. And I think that can help prevent some of this culling option when we can say that kind of early warning system, which I was talking, and that can be strengthened only by having a very good surveillance, not just by looking at the wild birds or dead birds, but also doing some strategic, hot spotting of the sites and looking for what are different kind of subtypes are floating in the poultry, but also in the domestic wild birds, but also the wild birds which are coming in the country. We do not have any such information. All we know where the outbreaks have happened and what's happening basically in those sites. So yeah, culling can be prevented if we have a system in place.
Yes, thank you. The next question goes to Dr. Uyeki, where the person has asked the importance of yearly revaccination in case of healthcare professionals in current scenario.
Yeah, thanks for that question. So, annual influenza vaccination against seasonal influenza, right, is recommended. I won't speak about India's recommendations. I'll just speak sort of on a global picture, but healthcare personnel are recommended for annual influenza vaccination. Now, influenza vaccination against seasonal influenza is not going to have any protection against avian influenza virus infections. And so while it's important, it's not going to prevent, if you have an H5N1 patient, it's not going to prevent transmission. So the key in hospitals is better infection prevention and control, and it's utilizing the recommended personal protective equipment. So for an H5N1 patient, we recommend fit tested respirators, eye protection, disposable gown and gloves. But we also recommend isolation of the patient. If the patient is going to have healthcare personnel come in the room, the patient should actually have a surgical mask put on them to help reduce source, it's for source control purposes to reduce exposure to the healthcare personnel. And in a lot of countries, unfortunately, family members are in the room helping take care of the patient. And in those individuals, they need to wear the same level of personal protective equipment as the healthcare personnel. I've been in many hospitals in many countries of the world, including in countries with H5N1 patients, and I have seen the parents or another family member who's actually in the room providing most of the care not having any personal protective equipment. We need to protect the family members, actually limit family members. If possible, don't have family members in the room. So I think seasonal influenza vaccination prevents seasonal influenza, doesn't have any impact on preventing zoonotic influenza, novel influenza virus infections. Thanks.
Thank you. I would now, this is the last question that I need to ask Dr. Tosh, which is regarding which avian influenza subtypes are most zoonotic in Indian perspective? What are the precautionary measures to take from not getting zoonotic avian influenza?
Mostly the H5N1 and the H9 low pathogenic avian influenza H9N2 and which is already endemic in India in poultry, and H5N1 there are sporadic infections. So these are the two zoonotic influenza viruses circulating in poultry, whatever from the surveillance data in poultry indicates. And protection is these are the like directly coming into the contact of the birds, or when you are processing it is majorly the biosecurity measures to be followed when handling the birds or the processed meat.
Thank you very much for your response. There are a lot of other questions in the chat box which we could not address. We tried to address some of them. We will take note of those questions and try to formulate answers to them. But we don't have your contact details, so we'll see how to send back the answers to all of you. So I would finally like to hand it over to Dr. Anoop for concluding the session. But before Dr. Anoop chips in and gives the vote of thanks, I would personally like to thank Dr. Anoop very profusely for organizing this webinar, coordinating with everybody. So my sincere, heartfelt thanks to you, Dr. Anoop, and over to you for a formal vote of thanks.
Many thanks, ma'am. Many thanks for the opportunity. I would first like to thank the Office of PSA for bringing all the sectors together in our country, so that everybody has started to work hand in hand, shoulder to shoulder, leveraging our strengths so that we can build our nation based on a One Health approach. So thank you to the Office of PSA, Dr. Sindura and Dr. Nidhi, for all your efforts in getting this webinar too. Also, I would like to mention our second partner here is CDC India, Dr. Shailaja, their consultant, Dr. Mayank, Dr. Runa, and Dr. Siddharth, and Dr. Tim for lending his time. And I know it's very early in the day today for Dr. Tim to be joining us today. So thank you for your efforts and time. Also, Dr. Farah and Dr. Shailesh and Dr. Tosh, many thanks for accepting our invitation so gracefully and for enlightening us with your presence and talk. I also would like to thank all the participants who we had around 740 registrations for this webinar. And it's a huge encouragement for us to see all the questions and the discussions here. Really, I thank everybody and hoping to see more of such talks and cross-teaching across sectors in the future. Thank you, everybody.
Thank you.
Thank you very much. Thank you.
Thank you.
Thank you.
Good.